Okay, it's recording now. So again, welcome everyone. So today we have Professor Matthew Powner from University College London. So to introduce Matthew. So Matthew obtained his master's degree in 2005 and his PhD in 2009 in chemistry at the University of Manchester. Then he completed an EPSRC doctoral prize for postdoctoral fellowship with John Sutherland, and then he was awarded a Harvard fellowship to work with Jack Szostak at Massachusetts General Hospital. In 2011, he joined UCL, University College London, as a lecturer in the Department of Chemistry, where he is currently Professor of Organic Chemistry. So he has been awarded various prizes and fellowships in recognition of his research, including the Stanley Miller Award in 2011, the Roscoe Medal in 2012, Origins of Life Challenge in 2012, which is a joint prize. This was a first prize joined with John Sutherland, an EPSRC Early Career Fellowship in 2013, a Thin Chemistry Journal Award, I don't know whether I pronounced that correctly, in 2015, the RSC Harrison Meldola Memorial Prize in 2019, and he was a Blood Bannock Awards UK finalist in 2021. So thank you very much, Matthew, for joining us today. And whenever you're ready, you can go ahead. Thank you.
Thank you. No problem. Did you start recording? I don't know if you did. Good. Okay. Okay. Well, thank you for the invitation and the really kind introduction. So, as outlined in the introduction, I am a chemist, and I recognize that the audience is much broader than chemistry. So hopefully I won't go into too much complexity for how the reactions that I want to describe work, more hopefully a broader theme of the concept that I think is interesting here. But my group at UCL work solely on one challenge, and that is, if I can just get a pointer, is to try and understand a specific period in history when on the very early Earth, something happened, some chemical event happened that led forwards over billions of years to where we are today in Earth history, to lead to life and life as we know it. All life on Earth has sort of this, all the life that exists on Earth now has the same kind of historical record. It's all related to one another through inheritance, and it's all part of one family tree. What we want to try and understand is why specific chemical reactions of the types of compounds that could realistically be available at the dawn of life could have led to this form of life that we observe today on Earth. This is a really challenging problem for lots of reasons. And some of the reasons that it's a challenging problem are the reasons that we really like this area and really like to apply mechanistic organic chemistry to this area. So when we think about the history of life, we often use the fossil record to interrogate the changes in life and over its history. As we go back through the fossil record, we can find fossilized remains of organisms, and we can look at how those have changed over time to interpret the changes in life back in Earth's history. The problem for interrogating the origins of life through the fossil record is the further back in time we go, the less clear the fossil record becomes. So the fossil record is lost through weathering. And once you get back to the time period where life originated, about 4 billion years ago, then you're just running out of fossil record. And this places sort of an informational horizon which you can't see beyond for interrogating the origins of life. A second way we can interrogate life and life's history is by comparative biology and comparative genomics. And this lets us build phylogenetic trees, a simplified version of such is shown here. And this lets us compare the information that's in cells. So for example, the DNA within different cells, to see how it differs across different organisms, and then use that to sort of delineate a route back towards the historical ancestors of modern life. The problem with this process for, again, interrogating the origin of life is this process of comparative biology requires cross-referencing of ever wider, ever more different species to push you further and further back into history. And we just don't have enough species. We don't have any outgroups. Once we get back to essentially the difference between bacteria and archaea and eukaryotes, we're done. This is the maximum extent of our tree, and this pushes as far back as those domains universal ancestor, but we can't go beyond that. We have no extra information. Again, it places this sort of informational horizon that we can't see beyond, but we know the origin of life is beyond that last common ancestor. And that last common ancestor is really complicated in terms of its biochemistry, and it's almost certainly much more closely related to modern life, to sort of the complexity of a modern bacteria, than it is to the first reactions that eventually led on towards life. So this gives us a really big informational gap between, actual chemistry that initiated this process and the furthest back we can drive with comparative biochemistry. And this means that with both the fossil record, so the geological record of what the Earth was like at the origin of life, and the biological record of modern life, there's just insufficient constraint to know the chemistry that led to cells that are on the Earth today. And we could envisage lots and lots of different circumstances of types of reactivity that could lead to compounds, and we can interrogate whether these look like living or life's essential molecules or not. And here's a few listed. We could keep listing them forever. There are so many different potential circumstances for chemistry to happen across the diversity of environments that are available on a planet, that there is very little scope to narrow this down to specifically say this is the environment that gave rise to life. And none of these chemistries that derive from these kind of environmentally constrained reaction sets really look like the biochemistry of life. There's something more going on here. This doesn't constrain the types of chemistry that we need to build the molecules of biology to actually give them. So what's missing? What else can we do? So this is where we think organic chemistry comes in, and this is how we approach this problem. So we approach this problem not through geological constraint, but through reactivity. We want to understand how the reactivity of simple molecules could specifically lead to the molecules of life. So you can envisage lipids, you can envisage proteins, you can envisage DNAs, RNAs, metabolites, cofactors, all of these compounds that are universally conserved across life have the deepest rooted history in life's chemistry. And we want to understand whether there are reasons in basic organic chemistry and mechanistic understanding of the reactivity of simple molecules to specifically derive these compounds. And if that was then true, can that back inform our understanding of the environment from which these molecules could have arisen? So where I want to begin the discussion of our chemistry is this paper. This paper came out at the end of 2020 and is really a perspective piece on thinking about the origins of life. But it sets this up in a really unusual way for me. And that is thinking about water as a paradox. This seems to be a prevailing thought in the field. And it's brought together in this article, and lots of different people involved in origins of life chemistry are in this article. But and this is just an example of how this has been cited. But what's being portrayed here is that water is some way not required or will be counterproductive in our search for the origin of life, and that because we envisage that, because we've encountered problems with the chemistry that we would like to do in the context of it working in water, then we will throw the water out. We will assume that water shouldn't be there and life should begin without water. And here's, a great kind of summing up of that process, where someone's suggesting that water could be viewed as a poison to the origins of life. And this makes literally no sense. There is no paradox. Water is fundamentally essential to the chemistry of life, and water is fundamentally a part of life's chemistry, and it will dictate how the chemistry will work and how chemistry can work. And I think what we need to consider here is what we're assuming when we allow ourselves to remove water from this system, and what the problems are that water gives rise to, and whether these problems are intrinsic problems or problems because of our perception of how we choose to do the chemistry that we think will lead to the origins of life. So maybe what we should consider is, if the chemistry doesn't work well in water, we're actually pushing in the wrong direction. And that might be a really key clue to unraveling how these molecules arose and why these specific molecules arose in originating life. So basically, reposing that question of should water be removed as, well, if water is essential, does this focus our search for the reactions that originated life? And to think about this in the context of actual molecules, I want to try and present two different pieces of work that I've been involved in that both talk to this question of is water a problem, or are we thinking about the reactivity from the wrong angle? So the first is some older work which looks at the nucleotide synthesis going through oxazolines rather than sugars. And then the second is newer work which has related overtones, which is why I've put these together despite the 10-year gap between when they were done, for peptide synthesis, and whether we can learn the same lessons and whether we can think about water in this context, and how we should think about the chemistry we use to build molecules in the context of the origin of life in an aqueous environment. Okay, so nucleotides. These are complex molecules, and for non-chemists, it's not super important that you know the total structure of these, but what we're going to look at really is this first one here is, can you synthesize? This is cytosine. It's a complex molecule that allows information transfer in biology. It's at the root of why biology works. Without this, there is no biology. It's complex because it's made of lots of different chemical elements that must be positioned and placed together in a very specific way. So we have a phosphodiester backbone with a very specific ribose sugar that has very specific stereochemistry, has a furanosyl ring, and has a set of nucleobases attached to it via a very specific nitrogen. And if you get all this right, if you get all this structure right, then you can transfer genetic information. This becomes the molecule of biology. But there are so many ways to get this wrong. There are so many different sugars we could use. There are so many different attachments of how it could be ligated together, even just with phosphate. All of these nucleobases can be attached differently to the sugar. And there are so many other nucleobases that could be attached. So really a question that drives us here is, why would this molecule, of all the millions and billions of potential isomers and homologs, be selected? Or why would this one be present at the origin of life such that it could seed evolution and genetics? So where to begin? So the place that everyone has begun thinking about this molecule is a very obvious place, that it looks like it's made of two parts. It looks like it's made of this nitrogen-rich base, and there's 4 of those, and this oxygen-rich sugar, which are then strung together with phosphates. So this is a really obvious place to start, and with really simple molecules. So here's cyanamide and cyanoacetylene. These are really simple, abiologically plausible molecules. You can follow a trajectory of reactivity where these react and then react together to build a nucleobase like cytidine. So that works, and that's a positive for this thinking, that we could make nucleobases and then attach those to sugars. You can also take simple sugars and react those together to make more complex sugars, and this is the sugar we want, ribose, which could be made from a 2-carbon and a 3-carbon sugar. It has 2 forms. We want the minor form to then couple to our base to make a nucleoside, and this is where the chemistry really falls down. So this chemistry really doesn't work and cannot work in water. We're trying to attach an unactivated sugar, a very unreactive substrate, to an unactivated nucleophile, removing water to build this glycosidic bond, and this reaction is really very, very poor. You can dry it and you can push it and you can heat it and you can add salts. It's still very, very poor. You can make it sort of work a little bit, but not very well. And we're fighting the natural reactivity of molecules then to make these. And at this point, we have to question, why are we doing this? We've made an assumption about how this molecule could be made. We've tried it and it works very badly. And we are forcing the compounds to react with ever more energy, drier conditions, more extreme conditions. Why? Have we assumed something wrong maybe? Have we thought about this in the wrong way? We're trying to make the compound from something unreactive because it's aromatic, and something unreactive because it's not an electrophile, something that's a minor component of an equilibrating mixture. The reactions to make these are really high pH. We're talking sort of drain cleaner, pH 14 basicity to make this reaction work. Once we make this reaction work, the products are totally unstable to the conditions that make them. These are just as easily deprotonated and isomerized as the original input molecules. If you look at what we're overall trying to do, is we're trying to couple 2 elements that we think build this nucleoside by taking water out of these unreactive substrates. But if you look at our synthesis overall, we put this water in ourselves as we began the synthesis. So we're adding water, deactivating the substrates to take that water out. We're not using the high energy nitrile bonds here effectively. So is there another way? Have we assumed things that create problems for us? Just assuming we have to make this molecule via one route from sugar and base, does this create more problems than it solves? Another element to the assumption that we glibly make is that these nitrogen-rich and these oxygen-rich bits could come from separate chemistries. We assume we can get them from separate places. If you order your chemicals from Sigma Aldrich, then of course you can get them from separate places. But environmentally, we want to unite these 2 structures we should really want to build them together in the environment that we want to unite them within. And ultimately what we've done is we've seen a structure. We've looked at the constitution of a molecule and decided how we're going to make it by its structure, not by the reactivity of the substrates that we want to build it from. And that just doesn't work. That's not how you design chemical reactions and chemical syntheses. So here's another way. If we take the same substrates and we unite them now, the nitrogen oxygen rich molecules together, we can drive this molecule, this bond here is the bond we couldn't make. So we can use aromaticity to drive the formation of this bond, uniting nitrogen and oxygen molecules from the start. This is now high yielding, really effective chemistry that uses the energy in our triple bond to make the compounds that we want. This also locks this compound into a reactive substrate. This hydrogen here that we had to remove with extreme pH is now already removed, again, because of this aromaticity. It's locked in this reactive enamine conformation that allows it to react with the sugar to deliver 5-carbon sugar with complete furanosyl selectivity. We don't have this furanosyl-puranosyl problem now. We only get the furanosyl structure that is embedded in natural RNA. Again, by making this structure again, we lock the intermediate into a reactive form. We lock this nitrogen now into a form that will react readily with, again, one of our input molecules. So this nitrogen is really reactive towards our input substrate that can build the pyrimidine base, whereas the urea nitrogen was really poorly reactive. So this was a difficult reaction. This now is a facile quantitative reaction that works incredibly well to deliver this product and only this product in really high yield. And now over these 3 steps, the chemistry is being directed by the intermediates that we build to specifically give us the molecules we want and give those in high yield, rather than having to try and force the reactions with heat and high extreme pHs. So again, once we get here, again, we've built energy into this structure. This is now not a nucleoside, it's an anhydronucleoside. So that dehydrated nature of it allows it and drives the reaction of it with phosphate to deliver the natural component of RNA. But no, we didn't dehydrate this compound. We didn't take water out of it. We never put it in. The activation to build the phosphorylation into this compound comes from these input high energy nitrile substrates that enter our synthesis at the beginning. So the key here is that we're not wasting the energy in these high energy molecules. We're using them to design and to deliver syntheses that specifically give the reactivity that can lead to nucleic acids. This is not a geochemical synthesis. It was never designed to be a geochemical synthesis. It was designed to be an evaluation of chemistry. How can this chemistry work? Why would it give us the specific molecules that you find in biology? And using that, then you can go back and think about how would you constrain this chemistry in a geological, in a prebiotic context. But it's thinking about the chemistry differently and learning from the reactivity of molecules to think about how to deliver the molecules of biology in an effective strategy. So moving on from that, I want to think about a different class of molecules, but obviously one that are united with nucleic acids in biochemistry, and that's peptides. So again, we need to think about where to begin. So nucleic acids are fundamental to life. They're fundamental to information transfer. The information in them will ultimately be passed into sort of the workhorse of the cell, the thing that will catalyze the reactions, provide structure to the cell, and that's peptides in the form of proteins. So again, where should we think about beginning in building protein molecules, peptides. So this is the simplest form of peptide that I can show. This is a fundamental structure that is important to biology. Where does it come from? Why is it there? And why does it look the way it looks? Biology's protein molecules, peptides, all look sort of the same. They have an amine and an amide that then form a polymer that are each connected to one carbon. So they're built formally from alpha amino acids. And there's 20 of these, 22, depending on how you look at the genetic code, that are universal to all life and are used to build all protein molecules. So the question we wanted to ask was why these? Why these specific molecules? What is it about the chemistry of these that allows them to sort of seed the structures that will become essential to biology? So this overall is the chemistry we would assume we want to do. We want to take amino acids, alpha amino acids, and observe them react to build peptides. But again, this is very much like the reactions we were thinking about with nucleic acids. What we're wanting to do here is take unreactive structures and have them condense in water to deliver a peptide bond. And this really doesn't work at all very well. Not surprisingly, I think, to anyone who's studied chemistry at any level. If you take an amino acid, alpha amino acid, as would be found in biology, and put it into water at physiological pHs, then the amine protonates and the carboxylate deprotonates, we get a Zwitterion that's really stable and unreactive as a nucleophile and unreactive as an electrophile. This is, again, like our glycosylation bond, difficult, not achievable in water. How do we get around this problem? Do we dry it? Do we add more salts? Do we heat it hotter? Do we try and force this chemistry to happen? Or do we consider, have we made a mistake in how we analyze how we should get to this compound? This is the compound we want. This is the compound that's essential to biology. The assumption we make is that we should build it from amino acids. Is that the right assumption? Well, the amino acids aren't well designed and not well, the structure of them is not congruent to them forming this bond. To make this amine reactive, we'd have to do it at high pH. To make this carboxylate reactive, we'd have to do it at low pH. We could alternatively add activating agents, try and activate this whilst we do it at high pH. But this then brings in a whole set of problems for how we don't activate and don't react on all the different R groups, all the different side chain, reactive side chains that amino acids have. Are we trying to solve a problem of our own creation here, though? Do we need to make peptides from amino acids? Is this an assumption that we're making? Well, then we can think about, where do the amino acids potentially come from? So amino acids in the context of origin of life, abiotic chemistry, could very easily come from the high yielding Strecker reaction. If we take simple aldehydes, so formaldehyde would give glycine, for example, ammonia and cyanide, they will condense together in water in really high yield to make a stable compound, an aminonitrile. This aminonitrile then has to be hydrolyzed. We have to put water in it. We have to deactivate it to make the amino acid. This reaction is extremely slow and it doubly deactivates our structure. Not only are we feeding water into it that we have to take out of it by reactivating this carboxylate group, we also really change the structure of the amine in a fundamental and important way. This amine, the original product of our prebiotic chemistry that can potentially synthesize amino acids, has a really important pKa. So the pKa of an amine is, or its pKa is the pH at which, or it defines the pH at which it will be 50% protonated in solution. So an amino acid has an amine pKa of about 10. So at any pH that biology should work, so near neutral pHs, it's going to be protonated, its reactivity is quenched. The aminonitrile, because of the difference in the nitrile and the acid, this massive electron withdrawing potential of this nitrile drops this pKa 4.5 orders of magnitude down to 5.2. So any neutral pH, the aminonitrile is set up to be a nucleophile. It's predisposed to be reactive under biological conditions. And these are the conditions that really are constrained by biochemistry. To make Watson-Crick base pairing work, we have to be within these constraints. This isn't an arbitrary constraint of geochemistry or our decision of how we do the chemistry. This fits in the context of how the cell works, how life works. To have Watson-Crick base pairing, we have to be in the deprotonated range for A and C and the protonated range for U and G, so that we have to be between about 4.5 and 9. This is where chemistry can work in the context of biochemistry. So was it our assumption of how we build peptides that were wrong, or was the conditions wrong? Well, are we trying to do something by uniting amino acids that is counterintuitive? We're essentially trying to take an aminonitrile, deactivate it by hydrolyzing it, and then reactivate it to make peptides. Would it make more sense to come straight from the accessible high energy structure directly to the intermediate energy structure, the peptide that is functional in our biological context? And this, I've obviously set up in a very specific way, works incredibly well. So if we take an activated peptide that we want to grow and try and add an amino acid to that in water, this is a very poor reaction at neutral pH, whereas the addition of an aminonitrile is an amazing reaction at neutral pH. It works incredibly well. Addition of our aminonitrile, and I'll come to why this is a sulfur in a second, to an activated growing peptide works beautifully and is set up to work incredibly well because of the aminonitrile structure in under physiological constraint. Once the aminonitrile is added, so here the nitrile is activating the amine to be a nucleophile to add to the peptide. Once it's added, it's not an aminonitrile anymore with an NH2 here, it's an amidonitrile. So this goes from being an electron donating group to an electron withdrawing group. So this activates this nitrile relative to the unligated nitrile and allows it to be converted back round in a cycle to an activated species again through addition of, again, a nice, simple, geologically plausible substrate, hydrogen sulfide, sorry. And we can then set up a cycle where the nitriles, the thermodynamics are driving this chemistry, but it's not so reactive that it will react on its own. It won't just polymerize, it's controlled, it's kinetically stable, but thermodynamically reactive. And this is really important in terms of making this chemistry efficient, because this gates the chemistry. Each of these steps stops. This means you can drive them to completion and you can get high yielding defined products that you can't get out of high energy activation chemistry of polymerization of amino acids. This chemistry works across all of the proteinogenic amino acids really excellently. And I want to just look at one of these. So lysine, this is an important amino acid, and it's a difficult amino acid to work with in the context of abiotic chemistry because it has two amines. If we take an activated growing polymer and try to couple lysine here with a carboxylate at X, or lysine amide, this could be a peptide or it's just simple amide, we get little or no selectivity for the blue nitrogen, the one that we need to couple to make proteinogenic peptides, over the side chain nitrogen, the side chain amine of lysine. But if we use an aminonitrile, this is totally different. So because of this electron withdrawing effect, because we've dropped the pKa of the aminonitrile amine by 5 orders of magnitude over the side chain amine, then this chemistry is totally selective with nitriles. No protecting groups needed. The water that we do this chemistry in defines how the selectivity will work, and it directs the natural selectivity to give the proteinogenic amino acid ligation that we would need to build proteinogenic peptides. So the final point about this being, and I guess my final point I'd like to make to you guys here, is that by making this, defining this chemistry as kinetically stable, so inhibiting just the polymerization of these reactions, this importantly makes this reaction scheme amenable to catalytic control. So that with catalysts, you can now define a catalytic cycle to undergo peptide ligation, where the catalyst controls the reaction. So like you would have a polymerase catalyst that works in the context of biology. And now with catalytic control, you can take these same substrates, aminonitriles that we built through the, amidonitriles that we built through our aminonitrile coupling chemistry, and in high yield, dilute solution in water, selectively ligate these amidonitriles to amine nucleophiles to build peptides in high yield, in dilute aqueous solution with no water problem. If we take the amine coupling partner out of this reaction, so we just have catalyst and nitrile, there is no hydrolysis. We don't see background hydrolysis in this reaction. We just see reversible addition of the thiol until the amine engages to deliver peptide synthesis. So that's where I want to end. And you know, if you're a chemist, I apologize that I skipped over lots of the detail of how the chemistry works. But I recognize we have a very broad audience here. Really, the point I want to make here really throughout this talk has been there is no water paradox. Water is a key element of life and should be essential, and an essential clue to how we go about investigating the origin of life and defining the chemistry that could have led to the origin of life. And that we need to work with the reactivity of compounds, the compounds that will deliver these molecules, not against them. If we find problems with the synthesis, this might be an indication that we're going about it the wrong way. There may be an easier way, a facile way to make the compounds that we want. So I'll end there, and thank you all for listening, and happily answer any questions that you guys have about our chemistry.
Thank you very much, Matthew, for this very nice talk. So are there any questions? If not, so then I'll ask first. So if you have any questions, you know that you can also put them in the chat, and then I'll read them loud. But then to leave some time for people to write them. So I have a question, which is, so for this second part of the talk, you mentioned that you start producing, or you may start producing the aminonitriles from HCN, no, I think it was, and from ammonia.
Yeah, I go back to that.
Yeah, then, but for the production of ribonucleotides, so there's one key molecule in that scheme, chemical scheme, which is glyceraldehyde.
Yes.
And glycolaldehyde, I think. No.
Yeah, I can go back to that. Yeah. Yeah, here, these two.
So how do you produce those in the first place?
Yeah, it's a great question. So there are many potential ways one could consider this, and the one I like comes from, we've not specifically worked on how to make these compounds in my lab. We did publish a paper in 2017 about how to separate these out. If you had a big mixture of sugars and you made many sugars, how you could selectively separate these from that mixture to deliver this chemistry. We published a paper in 2017 in Nature Chemistry about this. and that uses the fact that this compound can isomerize to dihydroxyacetone, and this one can't isomerize to anything else to deliver selectivity in separating those. But that's not how to make them. How to make them, the synthesis I particularly like, it's not ours, it's one that comes from my previous supervisor, the Sutherland group. They demonstrated that you can make these compounds, both of these, from hydrogen cyanide. So the compound that we used in the Strecker reaction in the second part to make aminonitriles. And there are multiple ways sort of they have of doing this now. So I'll just won't describe the details, I'll just sort of explain how it works. They take hydrogen cyanide and an electron donor that will be excited by UV light. Hydrogen sulfide's a good one, for example. The UV light then causes the sulfide to release an electron. Sulfide gets oxidized and the hydrogen cyanide gets reduced. This gives you formaldehyde, and formaldehyde then reacts really quickly with cyanide to form a cyanohydrin. So it's a bit like our aminonitrile here, but where this nitrogen, so if you take this ammonia out, you get an oxygen here. And this compound, again, if you go through that same process of delivering UV photon, ejecting an electron from a stoichiometric reductant like hydrogen sulfide, this can then reduce this nitrile with an oxygen here, and then you get that compound, this compound. And then you can just go through that process one more time, add cyanide, give electron, reduce, and then you get this compound and ammonia as a byproduct. So you get the sugars, and you get sort of ammonia that I guess we need here. So there are processes. Is that the only way? I don't know. Probably not. There's almost certainly others. But what I like about that chemistry is, again, it works under these mild conditions because the addition of cyanide's a really amazing compound. Because the addition of it to aldehydes is so facile in water, and the equilibrium constant is so far towards product, this chemistry works facilely in water and delivers this sort of linear set of sugars and just exactly the small sugars that you need to set off this chemistry here. Does that help?
Yeah. Okay, so we have a question from Hector. Hector, go ahead.
Hi, great talk.
Thank you, Victor.
So my question was, in these studies they do from asteroids and comets, trying to figure out where the compounds for life were delivered from. There are many announcements like, oh, we found amino acids or nitrogenated bases. So are these other compounds also found in asteroids and comets so that they could be delivered to the prebiotic Earth?
Yeah, great question. So I think the, I don't want to say anything negative about anybody else's work, and looking for compounds in meteorites is obviously very difficult and very important for lots of reasons. But I think the question comes is, which compounds do you want to deliver to seed life? Do you want to deliver the actual molecules that we're assuming you build molecules of life like proteins from, like amino acids, or do you want to deliver their precursors that come with this energy? So the great thing about lots of atmospheric chemistry and space chemistry is that it does deliver these high energy molecules. So if you look out in space and you look at protoplanetary disks, for example, they are literally stocked full of things like cyanide in bulk, like oceans of these compounds that could, in principle, are they delivered? I don't know. That's well outside my sphere of knowledge. Could they be delivered from there onto the early Earth to actually build the molecules of life? I don't know. But from my perspective, some of the molecules that are found in these meteorites may well be sort of the byproducts of the inputs to life that didn't go into life. They've sat out in space without getting harnessed to build the molecules of life. And this is sort of the terminal product of that, right? So like I was saying, you can take an aminonitrile and you can, if you want, with effort and time, hydrolyze that down to amino acids, but you lost something in doing that, right? You took energy out of that. So I think it's a really interesting question to ask is, are those products that you find after you have investigated the meteorite, and you've got to think about how you get the compounds out of the meteorite as well. What did you do? If you're doing a really harsh acidic hydrolysis, how did that affect the compounds that you extracted? Is that, because that's exactly how you'd hydrolyze an aminonitrile to an amino acid. So is that product at the end sort of a smoking gun for the earlier compounds that had the energy to build life, or are those the exact compounds that you use to build life that you have to then put energy back in? And I don't know. I think it's very interesting. The thing for me in the context of the amino acids, for example, found in meteorites is there are so many, and so few of those that are found are actually the 20 that come into life. Then we need to overlay on that, you know, where did they come from? Why would all of those be present, and why would chemistry have selected out the few that end up in life as part of life? And you can see from the nitrile chemistry that I sort of described here, I didn't look at other analogs for you, but you know, this chemistry works, and activation works only because this nitrogen and this nitrile are attached to the same carbon. If I move it one further unit down, if I add another carbon in here, this all goes away. There's no electronic torque between these two elements. And so something like a beta aminonitrile won't do this chemistry in the same way. So wouldn't then become embedded in life, right? It wouldn't become embedded in the proteins through this chemistry, but it could be there in the overall molecules in the background, right? It just doesn't have that same level of reactivity. So I think when you look at the molecules that could be present, that's a good thing to do, but you have to overlay that on where did they come from? Were there precursors of equal or more value than the final molecule you analyze? And is there something about those molecules or their precursors that could determine which molecules react forwards and which ones sort of get left as sort of dead ends in a chemical sense? Does that, I hope that answers your question. I can't exactly remember what the question was now, and I've rambled a bit, but hopefully that sort of answers the question.
Yeah, thank you.
Thank you, Hector.
Thank you. Are there any other questions?
Yes, I have a comment or question regarding to this last answer. So what do you think, well, you will put the limit between the simplest precursors and the more complex species that you can form from these simple precursors? Yep. So I guess, I don't know if it's clear, the question. So I guess that what is the maximum complexity of the precursors that you think that will create the chemistry needed for the region of life?
Yeah, it's a very difficult question to give a specific answer to, because I don't like in the way you found it difficult to formulate the question. I think that's part of why it's a difficult question to answer, because there isn't a specific answer. There's no, you know, cutoff to what size of molecule gives you the origin of life. I think the key is, for me, in thinking about this, so as I said, I think about this not in terms of, you know, looking at what's in geological samples. I don't think about this in context of geological scenarios. I actually think about it in the context of how can the chemistry work? What will guide the chemistry to give us the bonds we need? Then I think what you need to do is think about all of the potential species. So if you're going to define compounds that you can sort of extract from, stick with meteorites. There's a lot, if anyone favors other scenarios, all are good, right? I'm just sticking to one scenario for simplicity of answering the question. If we're going to think about the context of the compounds you could deliver, right, then what you need to think about is every intermediate en route and whether they provide any value. So, for example, with hydrogen cyanide, you could deliver hydrogen cyanide, right? This is a compound that's very simple to make through atomizing any molecules that contain carbon and nitrogen, right? It's trivial to make in sort of dilute high energy chemistry. and things like Miller reactions, and in space, you see massive amounts of this compound, right? Because if you smash atoms apart, then they will recombine quickly, and they will give compounds like hydrogen cyanide, nitrogen, dinitrogen, and carbon monoxide, because this is the quickest way they can become stable molecules. You can take that compound then, and you can use it, right, potentially. Or you could then process it and use any of its products. So hydrogen cyanide, for example, could become formamide, or it could become ammonia and formic acid, right? So just through hydrolysis. That's a simple process. You could definitely envisage, you can probably find occurring in natural samples. But what you have to ask for each of those compounds, if you want to think about moving forwards, is which of them gives you the most value moving forwards. And for me, for those three, hydrogen cyanide brings something to the table that none of the others do. And that's a reactive carbon anion. If I want to build carbon-carbon bonds, hydrogen cyanide has chemistry that is specifically set up to be able to do this, right? It can bear a negative charge on the carbon of hydrogen cyanide in a way none of the byproducts of hydrogen cyanide could. So I think we have to keep a really open mind. Looking at what's there is really valuable, but we should also think, where did that come from? And could we have intercepted any of those precursors and got more value than the final thing we're looking at? So it's sort of a kind of, you know, it's a bit like doing molecular archaeology, where, you know, you see something at the end, which, you know, maybe a skeleton in the ground for an archaeologist, right? But you infer back from that there was a person there, right? And the person could have done all these things. It's not what you're looking at, but you have to take the story back in time and in context of where those things came from to develop the story and develop the maximum potential to learn from that. So again, I hope that sort of answers your question. And I think the answer to the question is don't draw a line at any of those points. Think about everything and use all the available information all the way back to atoms, essentially.
Okay, thank you.
Thanks for your question.
Okay, thank you. There's a question in the chat from Eduardo. He says, nice talk, sorry, no microphone. I have a question regarding the further oligomerization of nucleotides from nucleotides to RNA. In that sense, the OH, and in parentheses, bad living group, I guess, of the phosphate group is playing an important role for the phosphodiester formation. Do you think the prebiotic nucleotide is bearing a good living group, or might be in the cyclic form?
Yeah, good, very good question. And again, one that you could definitely investigate, and people are and should investigate from both angles. Both bring benefits. So the molecule that I left the RNA at was this cyclic phosphate, and this is activated with respect to a linear phosphate because of ring strain in sort of I've lost my pointer. Hopefully you guys can see my arrow.
Yes.
In this ring. Okay, so if we did have hypothetical equilibrium between this compound and polymeric RNA, it would favor RNA based on the strain in this ring. But that activation is very, very low and very difficult to make go forwards in this right direction. The way people approach this in the field generally is to move the phosphate here, which is where biology puts it, and put a really highly activated group here, like an imidazole, that can be then lost easily. This facilitates making oligomeric bonds for sure. I am not certain what is best here, and this is part of something that our lab is now investigating. So the first thing that I would note, putting the phosphate here is an interesting thing to do because biology does this. But if you take RNA, so if we're going to have RNA in a chemical system that's part of our cell, and that RNA is going to degrade and be recycled, like I'm going to assume that we're not going to use nucleotides once and then throw them all away. You know, they're high value substrates that a cell would like to reuse time and time again and can reprocess into new different information, just like biology does now. So if we're going to do that recycling process for RNA, we are specifically, RNA hydrolyzes specifically back to this compound. So because RNA in its polymeric form has a hydroxyl here and a polyphosphate across here, the way it hydrolyzes is to give this structure. So if we make RNA first time round, however we make it, we're going to come back to this structure 1. Right? That's inevitable for how RNA chemistry works. So then to come back to RNA by the 5-phosphate, what we're going to have to do is take the phosphate off totally and put it back on again, which with enzymes is fine. With prebiotic chemistry is really challenging. But so that gives me cause to think we should consider 1 very carefully. How do you get from 1 back to RNA? Also, I think in the context of what I discussed, I'm going to just go back off RNA because I don't have any slides for RNA, but I do have a slide for peptides, and it's related. So when I was talking about making peptides, which is now potentially building polymers, right, rather than the RNA chemistry, which is building monomers, what I said was of real value here to control this chemistry, to direct this chemistry, and to be able to define when and how this chemistry worked was that the nitrile here is activated. It has some energy, but it's kinetically stable. And this is the same thing with the phosphates. Okay, so you can have a high energy phosphate like a phosphorimidazolide, and this is really kinetically unstable. It will hydrolyze in water. Whereas if you take a cyclic phosphate or biology's activation, which would be a triphosphate, then these are thermodynamically activated but kinetically stable. They require catalysis to make them move forwards. So I think this is not a question I have an answer to, but it's something we're heavily investigating in the lab, is how much value is there in the difference of, there's 2 angles to this, really high energy phosphates that could auto-polymerize and lower energy phosphates that require catalysis to make them polymerize. Is there value in having those low energy structures? And those low energy structures then could be polyphosphates, they could be cyclic phosphates, but they require catalysis. They're like this nitrile. They won't just react with an amine without the correct activation catalyst, which in this context was obviously was the sulfide catalyst here. So I think, again, as with lots of questions in this area, it's not one I have an absolute answer to, and I think both sides of the question should be investigated, and I can see value to both sides. But my instinct is that catalytic control will be really important here in getting us from molecules, small molecules, nucleotides, to larger molecules, oligonucleotides, polynucleotides.
Thank you. He says thank you for the answer. And now Carlos Briones has a question.
Hello, Matthew.
Carlos.
Good to see you again. Congratulations for your talk and all your previous work in the framework of prebiotic chemistry. You know that I've been following your work for years. So it's very clear from your talk that water is essential for life. Of course, is indeed we consider as a prerequisite for life when trying to find life elsewhere. But in any case, this does not mean that reactions occur in bulk solution, as you know, of course. And this underlines the possible relevance of surfaces, and in particular clay interlayers and so on. So as these interlayers in particular can provide local environments that allow water removal, what's your opinion on that? After decades of investigation on clays, as you know,
yeah.
What's your opinion about the current relevance in the framework of the origin of life?
I think they are relevant, and we have a project running right now in my lab to look at clay catalysis, not to remove water, but to catalyze different reaction, and sort of providing an acidic environment rather than a dehydrating environment is probably it's probably low water content, which is probably why it's acidic right in there. You will know that better than me, I would assume. But so I think they are relevant, and I'm not necessarily saying you shouldn't ever look at dry reactions. We've looked at dry reactions. The phosphorylation I showed, for example, in the I'll go back to it. So everyone knows what I'm talking about. So this phosphorylation here works by taking 13 and inorganic phosphate, helps if you have urea, it catalyzes it, and just taking a solution of those and drying them. And then that drying process makes that reaction happen. So that's okay to me, that we can investigate those things. The problem for me is suggesting that it's a paradox that water should be there. I think we have to investigate, again, all of this. I think if a dry reaction brings something to the table, then we should consider it and we should use it and we should see how far it takes us towards biomolecules. The problem I have is when someone's trying something and it's not working and you're just forcing it hotter and harder, then that seems like the time to me that we should reevaluate and ask, did we take a step in the wrong direction? Is the difficulty of doing this pushing us to look back at the assumptions we made? And so what I really wanted to get across here is not that this can't involve dry chemistry, which this step is still dry, this bottom step here. This step is lost. This could have been a dry step. It just doesn't work very well. Taking this back into water let us rethink this chemistry and bring this through in a manner that didn't need dry chemistry. So my current thinking is if we don't need to do it dry, we can do it in water, fantastic. Maybe we've not investigated any of these reactions dry, but I don't see why lots of these reactions wouldn't work dry, right? They could, and in principle that may help, but they don't need to be dry. So I think it's worth considering still. I'm not saying none of the chemistry should be dry. I think clays can provide really important environments, and some of those could be relatively dry within them. I don't think it's helpful, though, to consider water as paradoxical or in some way, we should be looking for chemistry that works outside of water. I think, life is a process that does now happen in water, and the more chemistry we can get working in that environment will smooth the transition from abiotic reactions to biological reactions. And I would like that process to be, just linear, smooth process, right? And having more chemistry in water is better. But clays are great, and dry is not off limits for me. It's just not a paradox.
Yeah, I fully agree with you. Yeah.
Thanks for your question, Carlos.
Yeah. Thank you very much, Matthew.
Thank you. Are there any other questions? No? Okay. Then, well, thank you very much, Matthew, for joining us today.
It was a real pleasure. I'm, sad that I can't be there with people, but it's great that we can talk over the internet.
Yeah, maybe in the future, we used to invite people to come over and then give the seminars in person. So maybe in the future, when the situation improves.
Well, I hope to see everyone who's in the crowd somewhere at some time in person. Thank you all for the great questions.
Yeah, no, thank you for your talk. It was great. I really enjoyed it. Okay.
Take care, everyone.
Take care. Thank you very much. And then we see you all next week. Bye-bye.